The overall goal of this procedure is to determine, in a high-throughput manner, the relative proportions of amylose and amylopectin in potato tuber starch. High amylose starch is desirable due to its lower digestibility. Potatoes with high amylose starch have a lower glycemic index and higher fiber content. This technique is being demonstrated by Philip Smith, an undergraduate student at the University of Wisconsin-Madison. In our lab, we're interested in breeding for more nutritional potato varieties. One way we can do that is to increase the amylose content in potato starch. High amylose starch is more nutritional because it has a lower glycemic index. And in this assay, we have developed a procedure that allows us to look at a large number of samples fairly quickly, and it's a repeatable, high-throughput, inexpensive assay. This assay is a modification of an iodine staining procedure. It is a colorimetric assay, and it can be run using a 96 well plate. Fresh potato tubers are used for this method. Dice the tubers into approximately 1 centimeter cubes and discard pieces containing skin. A French fry cutter allows this step to be carried out efficiently. The cubes are placed into a labeled bag. We use nylon socks, such as those used in shoe stores. They're inexpensive and allow samples to dry quickly and evenly in the lyophilizer. The nylon bags containing the samples are placed into a paper bag and frozen overnight at -80 degrees C.